My pleasure to introduce our next speaker, Brian Brown. Brian Brown is assistant professor in the genetics and genomics department and a key member of the Immunology Institute. And his background is pretty diverse, but I think the key attribute that's most relevant to the talk is he is a technologist, a fellow technologist. He's really big into doing really cool things with technology. And I think what's really awesome about this particular project is that, you know, these technologies like CyTOF, single-cell seq, these things are very often being used by people to do screens, to solve surveys and things like that. So it's something about learning about the characteristics of the immune system. The way that we can really expand beyond this to learn about biology requires perturbation. And there's many ways of perturbing systems. One of the hottest ways that's been coming up recently is genetic manipulation through things like CRISPR. And Brian's going to be talking about a new approach developed out of his lab that I think completely revolutionizes this technique and is going to make it much better and more useful for everybody.
Thanks, Sadiq, for the intro. And I will say thank you to you and Laura for inviting me to speak, for organizing this great meeting. Sold out in a day. And actually, Miriam, I was in a meeting with Miriam. She's like, oh my God, he hasn't advertised it yet. He hasn't advertised what's going to happen. Advertises it the next day, sold out. So it shows how exciting this is. Yes, I will say just to start the acknowledgements that this work was done by two very talented postdocs in the lab, Ola Blauska and Maxime Denon, and talented technician in the lab, Benjamin Benzby. And we really couldn't have done this project without the help of Adib and the HIMC. So it really has to do with sort of the pieces that were here that allowed us to come up with the concept of this technology. So as Adipa mentioned, you know, one of the things that biologists are really trying to do, I mean, probably 50–60% of labs out there are trying to figure out, you know, what do the genes in the genome do? There are more than 20,000 genes just in the human genome alone. And we don't know what most of those genes do. We don't know the function of them. So we have, you know, some knockout mice and, you know, there'll be some annotation here and there, some siRNA experiments, but really comprehensively understanding what each gene does in different contexts, our knowledge is actually incredibly poor. And so determining that function is one of the major goals, if not the major goal, of the modern era of biology. And in order to solve this problem, we really need new techniques that allow us to do this at scale because of the number of genes and the number of possible conditions there are. So if you have 20,000 genes, you know, you can say, well, I can knock out each one, I can create 20,000 knockout mice. But what if you want to see, you know, how each one affects, you know, cardiac disease or affects response to drug, or, you know, it becomes really impossible to do that. And you know, there's also the limitation of using things like mouse models. So we have to switch to human models or fly models or other types of systems. So in a way, this becomes a problem of massive scale. And what I always tell people, and maybe some of you have heard this talk before, which is, you know, the excitement around CRISPR, we often, you know, if you read popular press, the excitement often gets centered around therapeutics. But within the biology community, we all know that the excitement is really that it provides us a new way to knock out genes very rapidly. And the reason why we're all excited about that is because we all want to knock out genes very rapidly and figure out what they do. Now, even trying to do this at scale runs into the problem of how can you do this even with something like CRISPR. So one of the ways that people have been doing this is using approaches like an arrayed technology. And by arrayed technology, what I mean is, you know, you would have a 96 or 365 well plates, you put your cells in each well, and then you drop a different CRISPR into each well. Well, even if you're a, you know, big pharmaceutical company with tons of resources, this problem is still untenable because you would have, you know, let's say 20,000 genes. Each one requires 3 to 5 CRISPRs per gene. And then if you want multiple time points or different drugs, you're at a scale that even that no one can handle at the moment. So where biologists have turned to is a very exciting approach, and this has been around now for quite a few years, but it's really taken off recently, is to use a pooled approach in which you work with all your CRISPR guides or shRNAs as a pool. And the idea here is what you do is you synthesize your CRISPR guides, and each guide is targeting knockout of a different gene. You synthesize it as a pool. You get back a tube, and that tube is full of guide or oligos encoding guide RNAs. You shotgun clone that into a lentiviral vector or retroviral vector, and then you make the vector prep as a pool. So at the end, you have a single tube, and that tube is full of lentiviral vectors, and each one encodes a guide RNA that targets knockout of a different gene. And then you can drop those vectors as a pool onto your cells all at once, and you can try and study the function using a kind of Darwinian selection approach. So what I mean by that is you can have a plate of cells here, you drop your vectors on it, and in this particular example, 2 cells got a guide RNA targeting knockout of P10, 2 cells got a guide RNA targeting knockout of MYC. You can apply some selective pressure, pathogen, drug, time. In this particular case, let's say we just did time. And then you can see that what would happen is, depending on what the CRISPR hit, you would have a change in the frequency of guides. So in this particular case, because MYC is an important oncogene required for cell proliferation, when the cells carrying the guide RNAs targeting MYC, they were lost over time, and the cells targeting knockout of P10, they were actually enriched over time because the cells that lost P10 had a selective advantage. Now the question is, well, if you do this as a pool, it's not obvious which cells have which guide RNAs. So the way that we figure that out is we take the cells, we extract the DNA from them, because the lentiviral vector encoding the guide is integrated in the genome, and we can actually sequence to figure out which guides are in these cells and at what frequency by using a pooled sequencing approach. So you basically take the guides, and I'll show you better in a minute, you PCR across where the guides are encoded, and then you deep sequence it. And if you see an enrichment or a de-enrichment, this tells you something about what the gene must have done. So it looks something like this: library of lentiviral vectors. You drop them onto your cells, you apply your selective pressure, you PCR across the target sites. So you're at the end, you have a amplicon, which contains a different frequency of each guide, and then you deep sequence. This is an example of what this experiment looks like. So this is a study that Ola and I collaborated on with Emily Bernstein. It was run from Emily's lab and recently published in Nature Communications, where we built them a CRISPR library, just like I showed you, targeting different epigenetic regulators. And so if we take these melanoma cells, excuse me, we drop the library of CRISPRs on them, and we just allow them to proliferate for 42 days, and then we sequence the guide RNAs. Basically, the frequency of each guide RNA, whether it's targeting any of the genes in the library, is the same at time 0 versus 42 days later. But now when Thomas applied a RAF inhibitor to the cells, then we looked at day 42 with just DMSO versus day 42 with the RAF inhibitor, and now all of a sudden you see that certain guide RNAs were enriched. And what this tells you is that knocking out one of these genes provided a selective advantage for the cells to resist the drug. And so you infer from that that knockout of that gene must have something to do with the sensitivity of those melanoma cells to the drug. Now, this screening technology is used all the time. People are using pooled screens. We give out libraries all the time. There are people here doing it, and people all over the world. And it's provided very interesting answers to many questions. But there's also a lot of limitations, and in particular, my lab has been doing this, those type of screens for at least 8 years now, and we've grown very frustrated with the limitations of the technology. So for one, most of the screens that are using that are kind of limited to looking at cell fitness. So, I would say the majority are kind of looking at, does this gene impact cell proliferation or cell death? And I would say maybe 70-80% or something like that. But not only are they limited to screening for fitness, but you're not even actually directly measuring fitness. What you're doing is you're inferring fitness. So in these screens, you never actually directly measure the phenotype of the cells. You infer it based on what the frequency change is from the guide RNAs. And that is very limited for many reasons, especially if you want to look at particular markers. And we're a single cell sequencing symposium here, so the other critique that we know from many areas of biology is that bulk analysis can be blinding in many ways. And in particular, for these type of screens, it's very blinding, and I'm going to show you one of the reasons why for CRISPR screens it's a particular problem. And actually related to that, in these screens, you need a really substantial phenotype in order to actually see a change in the frequency of something. And part of that is because of the bulk analysis. And again, I'll explain that to you in a minute. The screens are time consuming and they don't allow spatial analysis. So we're always basically taking the cells off a plate, or taking the tumor, disassociating and extracting the DNA and running it through. We had the idea a few years ago, which is, could we generate a system where we could actually detect the CRISPRs and in a way that we could still do phenotypic analysis and permit single cell resolution. And the idea we came up with, which really did have a lot to do with the fact of our proximity to ADEEP and the HIMC and the scTECH, was we could develop a protein barcoding based system which would allow us to detect all these vectors. Now, what I mean by that is, I'll show you in a second. So we've been using protein-based reporters for many, many years. The most famous is GFP. We love GFP. But the problem with GFP and other fluorescent proteins is they have a lot of signal overlap. So at most, you can maybe put 3 or 4 fluorescent colors together. And if you wanted to do, a library of 50 different vectors or 50 different CRISPRs, that just wouldn't be possible because we don't have 50 different colors that we could do that with. There's no 50 different fluorescent proteins. And even if you tried to mix those 3, you would still get maybe 9 or 12 different colors that you could possibly get. But we had a different idea, which was to create a barcoding system where we use linear epitopes as the detection system. And by doing that, we could then detect our barcodes using a high-resolution means like scTECH. And so what we did is we started with a surface receptor, NGFR, that doesn't have a signaling domain. So it's actually a mutated version. And we fused to it linear epitopes that could be recognized by an antibody. So these epitopes can be things like HA or FLAG, and we ran through many different ones. And so you could fuse 1, 2, 3 together, and you fuse them to this cell surface receptor for the reason that then when the receptor gets expressed, it exports these tags to the outside of the cell, so you could detect them there if you wanted. And then what you can do is you can actually reuse some of these tags. So procode 1 can be tag 1, 2, and 3, and then you can make a second procode which reuses some of the tags, but doesn't reuse all the tags. So in this particular example, we're reusing tag 3, but we have 2 new tags. You could say procode 1 is tag 1, 2, 3 positive, but procode 2 is tag 4, 3, and 5 positive. And using this principle, you can use this formula, which is a combination, not permutation, and looks something like this. So if you have 3 positions and 10 epitopes, that's 120 different possible combinations that you can create. If you take the same 10 epitopes but now use 4 positions, you can go up to 210. And if you have 20 epitopes recognized by 20 antibodies with 5 positions, you can create 15,500 unique proteins that can be potentially detected. So does this work? So Ola identified 10 linear epitopes that could be recognized by commercially available antibodies. And she conjugated them with metal, fused, created every possible combination of the 10 by 3, which is 120. And this is just showing you an example of 18 of those vectors. So we take 18 lentiviral vectors, we drop them onto cells at a very low multiplicity of infection, and then we stain with the antibodies that recognize those same linear epitopes. So there's only 10 antibodies in this cocktail, and then we go to the scTECH and analyze it. And this is just what the scTECH plots look like, just showing you the difference between the negative and the positive is quite good. And then we can actually use LAD's algorithm, the Visney algorithm, and you can actually cluster. So we don't even tell Visney how many clusters there were. It just basically spits out. It sort of looks for, what is the similarities between these populations, and it found all 18. And what I mean by that is you can see each of these clusters is containing single cells, and they're positive for a particular epitope, but each cluster is positive for 3 and only 3 epitopes. If it's positive for 2, it's thrown out. If it's positive for 4, it's also thrown out. So you can see here, if you take this cluster, it's negative for epitopes 1 through 6, but then it's positive for epitope 6, 7, negative for 8, 9, and positive for 10. So if you know which procode that was, so procode 5 encoded epitopes 6, 7, and 10, you would know that that cluster, every cell in that cluster is encoding that particular procode. And if you linked it to a guide RNA, you would know what guide RNA they're carrying and what knockout they would have. So this is using the whole 120. So Ola again went back, this is 120 different vectors, put it onto cells at a low multiplicity of infection. Each cell only gets 1 vector, then goes to the CyTOF and uses Visney to cluster, and you can see in every cell type we've ever tried it in, we're able to easily resolve all 120. So this is really the largest set of protein-based reporters that have ever been created, and we only need 10 antibodies to detect all 120. So if you're using CyTOF, that leaves 30 channels left for phenotyping your cells. Ol actually went up even higher, so she went up to 14 epitopes that she's created, and 14 by 3 gives you 364 possible combinations. She created all 364. We put them onto cells, and this is what the clustering looks like. It can also actually tell you if you're colorblind or not. And then this is the frequency across it. All right. So it works extremely well. The question that people always ask us then was, would this work in vivo? We took all 120. So most of what I'm going to show you is the 120. We dropped them onto breast cancer cells, and then Maxime put them into these breast cancer cells into a mouse to grow a tumor. He actually did this in both wild-type mice and rag mice, because we wanted to ask the question, not only could we pull out the tumors and then detect them, we also wanted to ask the question, were they going to be particularly immunogenic? And what you can see is basically that we have the same frequency of each CRISPR guide RNA, each procode, sorry, not guide RNA, each procode in the cells, in the tumors pulled out of either the rag mice or the wild-type mice, indicating there was no particular immunogenicity to the procodes or differential. And if you ask, why are these particular procodes higher than others, this is actually the same labeled here, but this is actually what the representation was in the pre-inoculation. So I won't go into the details here, but we're able to look at actually cancer cell clonality using this technology, even independent of CRISPR. Okay, but the technology that we find, or the application that we personally find most exciting, is the ability to now go back and do phenotypic screens with high-dimensional phenotypic analysis. So what Ola did was she created a library of procode CRISPR vectors. So each guide RNA was cloned into a vector that contained a different protein barcode, and we targeted something like 30 or 40 different genes. We created 96 different vectors, and amongst the 30 genes that we targeted were these cell surface genes, including beta-2 microglobulin, CD4, CD45, which would allow us to actually look at them quite easily by CyTOF, and that's exactly what we did. So we took this library, 96 vectors, dropped them onto cells. Each cell gets a single vector, and we put them onto monocytes, or THP1 cells, monocytic cells, that were expressing Cas9. And then about a week later, we went to the CyTOF and we stained for the 10 procode, the 10 epitopes, NGFR, which is linked to the epitopes, and 10 different cell surface markers. And then at the end of it, when we get the data, we put it into Visney. Visney clusters all 96, finds all 96 clusters, and you can see here. So each of these clusters is expressing a different guide RNA targeting a different gene. And you get this, crazy data set. So this is actually not even our biggest one. You get back 960 histograms. So each of these is in the columns is a different marker that we're looking at, and each row is a different guide RNA. So this is a crazy data set, and if anyone has LAD's number, then I can call them and can help us analyze it after this. But this is what we get, and if you go through, I'll show you in a more clear form. It looks like this. So here's the THP1. They all express CD4, and you can see that clearly here, except for these cells here, which are carrying guide RNAs targeting CD4. And we know that they're carrying those guide RNAs because they're encoding a particular protein barcode that we've linked to those guides. And you can see that even more clear here. And you can see this difference. It's going to be even more striking here. So this is another one. This is CD45. All these cells, this is a single tube reaction. All of these cells are expressing CD45. But you can see here, I'm zooming in, these are 3 different guides targeting CD45. This one, and this one. And you can see this is a characteristic difference in how well the CRISPR efficiency can work. So this guide almost completely eliminates, you have almost all CD45 negative cells. This guide here basically didn't work at all. And then this one had a mix. So you have some cells that are completely CD45 negative and these ones that are CD45 positive. Now, the reason why I want to stress this is because at the beginning, I told you one of the problems with bulk screens is that it's mixing everything up together. And one of the reasons why you need single cell resolution with CRISPR is because of this efficiency difference. So if we didn't have single cell resolution and we were just collecting all the DNA and throwing it onto the sequencer, we would be mixing these cells with these cells, and you wouldn't be able to discern that difference. So the fact that we can now look at each individual cell, this gives us a resolution that is very powerful, that even if the CRISPR efficiency isn't 100%, we can still resolve if there's a phenotypic effect of the gene knockout. This is just showing you, again, the wall of histograms that we get. We've repeated this. So what I showed you a minute ago were cell lines. Some people don't like cell lines, so we've also done this in primary human T cells. This is work that Maxime has been doing, unpublished. So he's taken Procode CRISPR library that he's designed, put them into freshly isolated human T cells, then he electroporates in Cas9 mRNA, able to express it. Once again, this is actually a small subset because he actually, we have the wall now of histograms will be 3,000. So this is a small subset of the data, and once again, you can see how some CRISPRs are extremely efficient. So these are CD4 T cells, you can see that here, all expressing CD4, but these guys that got the guide targeting CD4 have lost it. But this is really the first time, and I say this, you know, not just to promote the technology, but to say because it's super exciting for us that we can do, you know, pooled screens and get high dimensional data, get, you know, really a lot of phenotypic data. We've gone up to 19 different markers and 200 different CRISPRs. 200 different CRISPRs. So we can really do now, you know, really look at different phenotypes in a very complex way. This is just showing you a Disney plot. I'll scroll through this quickly. We've actually also done signaling, another thing that is not possible with current technology. Take the THP1 Cas9 and stimulate them with different cytokines, and when you do that, you get different phosphorylation of different stats. So I'll just focus on interferon gamma. If you give, if you give the cells interferon gamma, they phosphorylate STAT1, and we put a library targeting different cytokine receptors into the cells. Here's the Visny clustering. So each cluster encodes a different guide RNA, and this is just looking here. So we had guide RNAs targeting, these are the ones that are targeting the interferon gamma receptor. You can see we have very little phosphostat1 in those cells. But then if you look at single cell resolution, this is how it looks. So this is phosphostat1 in the THP1. This is it, the phosphostat1 after you give interferon gamma. And you can see here there are clusters that are darker than others. So if you look if you look carefully here, these clusters here are lighting up. So phosphostat1, stat1 is being phosphorylated, whereas these two clusters here, they have lots of purple dots because phosphostat1 hasn't been lit up. And that is because the CRISPR has a different efficiency, but you can see these ones are expressing Procode 1 and 2, which is linked to guide RNAs targeting CD116, which shouldn't affect interferon gamma signaling, and that's what you see, whereas these two clusters are expressing guide RNAs targeting the interferon gamma receptor, give interferon gamma, and they no longer signal through phosphostat1. Okay, so in the last part of the talk, I'll tell you a little bit about some of the applications we've been using the technology for, particularly for cancer immunology. That is what our major interest is, and where we're really trying to focus now. One, trying to find regulators of the T cell biology, but we're also interested in finding regulators of the cancer cell sensitivity or resistance to antigen-specific T cells. So for that, we use this JEDI technology that many of you are probably familiar with, that we developed a couple of years ago. These are T cells that are specific for green fluorescent protein loaded into MHC class 1, or peptide event GFP loaded into class 1. And this allows us to visualize the antigen in a powerful way. This is just an example. Some of you, I'm sure, have seen this video. So these are A20 cells, lymphoma cells. They're expressing GFP. These guys are also expressing GFP, but we've evolved them to be resistant to the JEDI. But you're going to see here in the culture. So these guys, the JEDI, are inside this culture, and you're going to see them latch on to the A20. You'll see there's a JEDI that's latched on to this cell right here, who is expressing GFP. Jedi is latched on here, and this is how the technology works. He punches a hole in him, and this guy dies, and this guy doesn't. So this is a way we can use the technology to study cancer sensitivity and cancer resistance to immune cell killing. We take these breast cancer cells and we engineer them to express GFP and Cas9, or RFP and Cas9, or no Cas9. We put the library of CRISPR vectors on them, and then we co-culture them with the Jedi T cells. And the Jedi T cells should only kill the cells expressing the antigen, their target antigen, which is GFP. And then we go to the CyTOF to look at what is the procode distribution in the cells. And we can also actually, as well, add markers to look at what's happening on the cells. So here's an example. Took these cells, they all have the library in them. The GFP and RFP, they have the same library. This is actually in the same dish. And we're able to cluster them based on the procodes. And remember, each cluster is carrying a different guide RNA targeting a different gene. And you can see here, no Jedi. We have about a 50-50 mix. We add the Jedi, and most of the GFP positive cells are killed, but the RFP positive cells are not killed. There's no Cas9 in here, so the cells are clustering based on the procodes, but no gene has been knocked out. Now instead, when we take the same cells, the breast cancer cells, we've engineered them to express Cas9, and now we put the Jedi on, you see a very different outcome. So here, you can see now all of a sudden we have cells that are resisting, as opposed to here with no Cas9. Once the Cas9 is in there, the cells start to resist because we've knocked out genes. Now we built this library in a way to sort of find genes that we knew were important anyway. And here's an example. So you can see in teal, these are clusters that are encoding guide RNAs targeting interferon gamma receptor 2, and in whatever color this is, these are guide RNAs, procodes encoding guide RNAs, linked to guide RNAs targeting beta 2M. And these are the resistant clusters. And you can see that even better here. Now, I have to say, when we first got this data, I was a little surprised that the interferon gamma receptor was even sort of a stronger hit, let's say, than beta 2M. Turns out that this is also being found in patients, and this has become sort of an important theme in cancer immunotherapy. This is a paper from Pam Sharma's group. This is one from Tony Rivas's group, one with PD-1, patients treated with PD-1, another treated with CTLA-4, and they both found the same thing, that a common reoccurring theme is that patients that are resistant to these drugs often have mutations in either the antigen presentation pathway, or they have mutations in the interferon gamma pathway. And this sort of leads to the antigen presentation pathway. That's less surprising to us, as we say, well, if you have no class 1, then of course the T cells aren't going to kill you. But it's a little more surprising, at least to us, that the gamma pathway was so important that it's like an actual resistance mechanism. And this had been described many years ago, that obviously interferon gamma was important. But what isn't so clear, and if you go into the literature, you say, so what exactly is interferon gamma doing? And sort of the hand-waving answer is usually, well, it's upregulating class 1, so it's all part of the same pathway. But in fact, in our experiment, we didn't see that was so much the case. And I say that because with the screening, we were able to add markers for MHC class 1 as well as other markers. But what we saw here, and I have to show you here, this is the MHC class 1 expression on T cells, which is already very high. And even when we put in a guide RNA targeting interferon gamma, we don't lose class 1 expression. Now, it's true that class 1 can't get upregulated as much, but they still express, these cells express a lot of class 1. And so this led us to the question, what else could interferon gamma be doing? And unfortunately, I don't really have a comprehensive answer for that. But the reality is, and this is the same with many interferon alpha, interferon gamma, they upregulate a very well-characterized gene expression program. Okay, so there are many genes, in fact, named interferon gamma stimulated gene, or ISGs. But yet we don't know what most of those genes do. I mean, if you go and look up, what does ISG XXX do, and we don't really know what its function is. And that speaks to the fact that we have so many genes in the genome that are poorly characterized. So we know interferon gamma is important, and we know it upregulates these genes, but we don't know what they all do. So Maxime actually said, okay, well, I'm going to build a library of genes that target all the genes that are, or many of the genes that are upregulated by interferon gamma. That's what he did. So he created a procode CRISPR library targeting genes specifically upregulated by interferon gamma or in the interferon gamma pathway, puts them on the 4T1 cells, and then puts the Jedi on them, and then went to the CyTOF. Now, when he went to the CyTOF, this is just the type of result that you can get from this, and I think this is super cool. Because when he went to the CyTOF, he said, well, I'm going to throw in antibodies. I'm throwing in antibodies to measure the procodes, but I may as well, since I have 30 extra channels, I'm going to throw in one to measure PDL1, one to measure class 1, one to measure whatever you want, basically. And so he had the PDL1 antibody in there. We didn't think it would tell us much. But when you look here, so this is PDL1, and you can see this sort of doesn't really do it justice. All the cells that in fact actually have upregulated PDL1 a little bit, but you see that there's 3 clusters here that have really upregulated it, and it's going to come out better in the histogram. Now, all 3 of those clusters encoded a guide RNA targeting SOX1, and if you look here at the histogram, so here's PDL1, and you can see that all 3 guide RNAs targeting SOX1, there's this shoulder here where you have uniform PDL1, but the cells with the guide RNA targeting SOX1, they all have even higher levels of PDL1, indicating that by knocking out SOX1, you actually get more PDL1 expression, and thus that SOX1 is a negative regulator of PDL1. And what was even more interesting is that it wasn't the same for class 1. So we saw no difference in those same cells, because you can actually go single cell resolution, in the same cells that had over-upregulated PDL1 and presumably have the SOX1 knockout, we don't see a change in class 1. And the reason why that's interesting is because you could have argued that SOX1 is just a overall dampening of the interferon gamma pathway, but actually this is sort of telling you that there's a separation in that pathway. And that's very important for cancer immunology because we want class 1 to get upregulated on tumors, but we don't want PDL1 upregulated on tumors. And a common thing that interferon gamma is doing is actually putting on the accelerator and putting on the brakes. So the fact that those pathways have molecular signaling components that are disassociated is very interesting in terms of what we'd be able to modulate. I just have 2 more slides, and this is actually why we did the screen, to look for things that would result in, if you knock them out, resistance to the Jedi T cells. So these are things that are downstream of interferon gamma. And 2 hits that came out, one was PSMB8, and another, which is a component of the immunoproteasome, and another was a gene called RTP4, which almost nothing is known about. It's actually a chaperone protein involved in chaperone and G protein coupled receptors. And this is just to show you better, because the screen and thing that we have to remember when you're using CRISPR and when you're using the screens is that the efficiency is not 100%. So if you see something like this, right? So what I'm showing you here, if we have no Jedi, we have an even mix of GFP and cherry cells. Now we add Jedi and we start to get killing of the GFP positive cells. I showed you this before, but if we knock out or we put a guide in targeting PSMB8, we see that, or RTP4, we see that some cells are resistant. Now you'd say, well, it's not that amazing a resistance, but remember that the CRISPR is not targeting all, not every cell gets a knockout. So if you actually then expand that population that survived, re-challenge them with the Jedi, and now they're basically 100% completely resistant. So losing RTP4 makes the cells completely resistant to antigen-specific killing. Even in this, and you could say, well, this is a reductionist model, but that makes it even more surprising because you have, just a kilo of Jedi cells in contact with the tumor cells, and they still can't kill them. This is the last slide. I'll show you. I just think this is a very cool feature of using CRISPR. Nothing to do with the ProCode technology, but a cool feature of doing these screens, which always frustrated us when we were doing these things with shRNA, which is you never knew if, when you had a resistant population or a sensitive population, or resistant usually, it was clonal. But with CRISPR, you can actually test that. So what Ola did is she extracted the DNA from the resistant cells and then sent it to be sequenced. And what you're looking at here, this is the RTP4 locus. This is, the arrow here is where the CRISPR, the Cas9 is able to cut. And you can see that each sequence she sent, or Sanger sequencing, looked slightly different. And so these were all different founding clones, different founding cells in the population that lost RTP4. So actually, it's not like it was like one cell type that just got enriched. These are actually different, it's a polyclonal population, and each one, the common theme here is that RTP4 was knocked out and they became resistant. So with that, thank you guys all very much. I just, again, want to thank the 2 postdocs who did almost all the work on the project, which is Ola Roblowska and Maxime Denon, and also to thank Adib and Miriam, and as well as Elad for all his help with the analysis and the vision. Thank you guys very much. take questions. No. You could do that's the HIMC's magic of having a good phospho panel. We were very fortunate for that. So yes, definitely you could do that. Yes, definitely. Yeah. I mean, as far as we can tell, I mean, the intensities of the clouds are pretty similar, which has something to do with the reagents as well, and titrations. But I mean, they should be an equal molar amount unless one gets swapped out, which is a possibility. Most of it depends on the affinity. What we always say, and I don't know, you would know better from BioLegend, but you have unlike mRNA, you have so much protein on the cell. There's probably like 50,000 molecules of the NGFR on the surface that the antibody, may be binding to an epitope on one antibody and another antibody bound to another one, and they may outcompete. We don't know. As far as we know, I mean, we're able to detect them quite well, and the intensity looks similar. I guess not the work. I would like to go to over 1,000, but I would say, and this comes down to, actually this is an interesting conversation for the group, which is, and we're not going to have it, but to think about, which is, it's the quantitative nature of these single cell studies is an issue. So if you go up so high, like if we were to create 10,000 procodes, then our ability to throughput that through the CyTOF is, the number of cells we would acquire becomes very low. And so how quantitative our marker identification would be reduced there. So if you only capture 10 cells expressing a particular procode, right, because you have so many procodes, then you don't get very good quantification of that marker. And I think that's a reality that I think gets glossed over, and maybe we won't hear, but that gets glossed over with some of the single cell stuff, which is, people treat 10 cells the same as they treat 200 cells, and they'll treat the quantification of those transcripts the same, and I think it's wrong. I mean, Effie would know better than me, but he's shown me some of the truth plots, calls them, where you have you have to think about the math of what your capture efficiency would be, and so I think you have to be very careful about doing too many cells and reducing the number that you catch of any one cell. Easily. We could go to 20,000. Yeah. Probably, yes. I mean, at some point it would be too saturating. I don't think we would do more than 5 or so combinations that we could do. Yeah. Thank you very much.